MPMI, which is the new virtual seminar series that MPMI, the journal, is hosting. We've been having, this is our third, we are having them every other week, and it's been a terrific way to connect with the community, to colleagues from all over the world, and has been a lot of fun for us as organizers. And I want to introduce my co-host today, Lijun Ma, who is also a senior editor at MPMI, focusing on genome sequencing and assembly questions, and she brings a lot of expertise to that. Really delighted to have her on the editorial board. And she'll be co-hosting, so we'll be answering the questions together. So brief comment on how we're going to do this. So after I get everything started, at the end, we'll be taking questions from the Q&A, and if you look at the bottom of your board, you'll see Q&A down there, and it would be great if you type questions into there. You can type them in at any time. We just will start answering them at the end. And look forward to hearing from everybody. We will stay and answer all your questions. This is a terrific way, I think, for people to interact with the authors. So one of the goals of this series is to help people engage and connect with MPMI, the journal, with our articles, with our content, with our authors, for colleagues across the world, and also to increase inclusion. And so we're trying to foster more of a culture of inclusion here by, this is freely available. You can access all of these talks. There's no subscription requirement. You do not have to be a member. That's true both for this talk right now live, and I'm delighted to see that we have so many people joining us live. We have over 100 already, but also the recording. So I know this is an excellent time for some people. We have chosen this time to be helpful for, to be a good time for, of course, the Americas, where I am, but also Europe, Africa, and the Middle East. This is still a good time. Although I know that there are people signing in from all around the world, and that's, you know, I really value your participation. Every other week, we'll be switching time zones. So our next talk will be at 9 p.m. Eastern Standard Time for the East Coast, which will be 9 a.m. in Beijing. And I've been writing a little Excel spreadsheet so I can keep track of this. So this will also be convenient for people in Australia and New Zealand, will be semi-convenient for people in India, will not be so good for people in Europe that time. And so that's why we're recording, because we really want to have everybody be able to join us. And we know that since we are a global community, this is the International Society of MPMI that hosts the journal, that we need to be able to adjust our time so that we can connect as well as possible. Also, because these are all recorded, you can go back over them at any time. And I think this is particularly helpful for listeners in our international community for whom English is not a first language. You can listen, you can go back and review parts that were a little hard to understand. And also you can ask questions of the authors themselves, something you can't do when you're just reading a paper. And so today, oh, and before I say that, so this is of course a fully accessible seminar series, and I just wanted to announce that MPMI will be going fully open access as of January 2021. Very excited to make that announcement here when we have our international community present. Really excited that we're going to be doing that, fully gold open access. Everybody will be able to access these papers. But I do want to highlight that currently all of the papers that are being discussed on this series, we are reopening them to open access so that they can be accessible. But of course, today's talk is a technical advance, and all of those are already open access. And so we encourage you, after the seminar, if you have more questions, to go back to read the paper, to dive a little more deeply, think about how you might be able to use this technical advance in your own research. And of course, you can always follow up by contacting John McDowell and David Haak, who are the corresponding authors, and you can find their emails on the paper. So, okay, so after that exciting announcement, and I should say that John McDowell was our last editor-in-chief of MPMI. Oh, I forgot to introduce myself. I'm Jean Harris. I'm the editor-in-chief of MPMI, and I'm hosting this series. Sorry about that. The previous editor-in-chief was John McDowell, who's one of our speakers today. And he really helped lay a lot of the groundwork for what we've been continuing to make the open up MPMI to be fully open access. So really excited to have him here. And so today's speakers are John McDowell and David Haak, and they're going to talk about the power of a collaboration. They are bringing different tools, expertise, and questions together to be able to really open up some new insights into the evolution of an important pathogen. And so I'm going to turn that over to them with one brief comment that our next talk will be by Hong Lu. And she's going to be talking about a very interesting virus-aphid plant interaction in which the virus actually gives the aphid a benefit when competing with other insects on the plant. So that's a really strange and interesting story. So that's going to be 2 weeks from now. Okay, reminder before we start, please type your questions into the Q&A box. You'll see it at the bottom. And Li Jun and I will read these questions and you'll get your answers at the end. Okay, so I'm now going to turn it over to John McDowell and David Haak. Welcome. Thank you very much. And of course, for people who want the speaker view, you can click at the top. There's a little box called Show Active Speaker Video, and that makes the rest of us go away. So, welcome.
Great. Thank you so much, Jean. And thank you for the invitation. And thanks everybody for joining us today. This is something John and I are both pretty excited about. I'll go ahead and share my screen here. All right. So yeah, today we're going to talk about nanopore sequencing of Phytophthora capsicum and how doing that revealed some hidden complexity. And again, I'm David Haak, and John McDowell is also going to talk a bit about this work. And you can see our contact information here on the screen if you want to get in touch if you have more questions after today. So as anywhere, we should start with some acknowledgements. And when I say we throughout this talk, I really mean these two guys, Chin-Ming Cui from my lab and John Hurley from John McDowell's lab. I put PhD here because they both subsequently have graduated and are moving on to bigger and better things, which is fantastic. So starting out, we had 3 simple goals for this project. The first was what I wanted to do. By training, I'm a plant and microbial genomicist. And what I really wanted to do is be able to resolve complex genomes in real time, right? To go to the field where pathogens were on a plant and actually sequence them, figure out who's there, and see if we can reconstruct genomes. A secondary goal is to see if we could use long-read data to improve the capsicum genome in its current state. And then finally, being able to just acquire the sequencing data is not enough, right? Even if we could acquire the sequencing data within 2 days, typical assembly approaches take 30 to 40 days of compute time with these long, noisy reads. And so we wanted to see if we could establish a more efficient computational pipeline for working with this high error rate data. And for an introduction to the organism, I'm going to turn it over to John McDowell, who knows that much better than I do.
Thank you, David. So as David mentioned, he entered this project looking for a complex microbial genome to serve as a test case. And fortuitously, we're 1 floor above David's lab, so he came round to our place and we said, boy, we have a good genome for you. It belongs to this organism that's called Phytophthora capsicum. So most of you, I think, have probably heard of the Phytophthora genus. It contains a number of oomycete species, many of which cause destructive diseases on crops. And in the case of Phytophthora capsicum, it's the worst disease problems it causes are on solanaceous species like peppers and tomatoes, cucurbits, and also legumes. the species collectively has a very broad host range and does a lot of damage on a worldwide basis every year. Now, the reason that we're particularly interested in this species is because there are certain strains of Phytophthora capsicum that grow very well on Arabidopsis. This comes from work that was pioneered in Francine Gover's group a few years ago. They did a very nice job of establishing an Arabidopsis Phytophthora capsicum pathosystem that was very easy for us in the McDowell lab to adopt. And we're particularly excited about it because it provides a very nice system for looking at root responses to oomycetes in Arabidopsis. And this was something that we previously lacked. And you can see in the pictures on the right-hand side of the slide the life cycle of the pathogen. Its spores attach to the epidermis of the roots, and then it penetrates to the inner layers and makes filamentous hyphae that grow in the cortex. Now, along the way, this pathogen secretes effectors into host cells to make the cells do its bidding. And the pathogen also acquires nutrients from the hosts that are through means that are currently essentially unknown. And so that in particular is an aspect that we'd like to explore. Now, as David will mention later, a very nice reference genome already exists for P. capsicum, but like most eukaryotic genomes, room for improvement exists. So we saw this as a great opportunity to set up a collaboration that would benefit not only my group and David's group, but everyone else in the world who's interested in Phytophthora capsicum or comparative genomics of Phytophthora species. So with that, I will turn it back over to David.
Great. So as was just mentioned by John McDowell, the Lamore lab produced a reference genome, which was a very high quality genome, especially for the time period, the 2012, when it was produced, right? So this assembly assembled a total sequence length of around 56 megabase pairs, just over 10,000 contigs, which from short reads is actually reasonably good. And with an N50 length, so 50% of the data was captured in 34 kilobase pair segments or larger. So this was a really good genome, but importantly, they estimated that the size with unassembled repeats was a bit larger, somewhere around 64 megabase pairs. Now, when we think about why they had unassembled repeats and why they could not get included, we have to think about how we assemble data. There's several different assembly approaches. For short reads, we most typically use a graph approach, and the most common is called the Brown graph. Through this, and most people are familiar with sort of the sequencing metaphor where we take a book and we chop all the sentences of the book up into fragments of uniform length, but their fragments still the same, right? So we have to try to reassemble those words and figure out what they mean. And so up here, I'm representing those short sequences as they come from the sequencer. And what we do is break them up into something called k-mers. Now, these k-mers can just be thought of as words, right? We're breaking up these sequences into words. And in this case of this example, I'm saying word length of 5. And of course, the k-mer length will vary depending on the length of the sequencing read and the quality of the assembly. But we can take these words and begin to identify how many of the same words that we've generated line up together versus how many unique words there are. And that's down here in the k-mer count. And so we get a distribution of k-mer counts. We can actually use that to estimate what our assembled genome size is going to be. But most importantly, we start to use these for our Brown graph construction. And here I'm showing you how we have these unique k-mers and they start to align together. But when we start getting into k-mers that have a higher count number, we start forming bubbles. And these bubbles get bigger and bigger until we can find algorithmically a path through those bubbles that gives us a reasonably good assembly. And when we do that, we call that resolving the bubbles. In some software, they call it popping bubbles. But once we're able to resolve those, we can get our sequence. Well, repeats, as you can imagine, will have a very high count number in our K-mer distribution. And when we have a lot of repeats, we can't actually find a path through them. And so they get held in that bubble. And so I've put over here, repeats are often held in unresolved bubbles. And so they never show up in our assembly. A strategy around that is to use long reads that can actually span the repeat or most of the repeat, in which we can use an overlap process for assembly. So here we have the same sequences again, but instead of breaking them up into K-mers, we're actually aligning them to each other and finding overlaps, and then we will stitch together the sequence based on those overlaps, generating a consensus call for the genome itself. Now, many of you have probably used Sequenture or some of these other approaches with aligning Sanger-based sequencing. It's the same basic principle, right? We are aligning multiple sequence reads to find a consensus for the full length of the genome. In this way, we're much better at resolving repeats because our reads are long enough to actually span those repeats. However, for some repeats, so in humans, some of the repeats are tens of kilobases, right? And so nanopore sequencing provides a great platform for trying to resolve those repeats because we can get sometimes ultra long reads. And so and the reason for that is the technology itself, right? So it is basically a pore that binds an enzyme that helps unwind the DNA molecule and a helicase, right? So it unwinds it, and then it splits it into single strand, and that single strand is passed through the pore, where the bases have hydrogens on them. Those hydrogens release a current as they pass through the pore, and that's what's registered. And this tracing actually gives you a really good example of why we have a high error rate, because you can see when you're passing through and you're reading the change in current, what we get sometimes is some interference. And it's interference from the next base that's to come through, or it's interference from an artifact in the system. But, and here's the sort of in real time process, right? So we're splitting the double-stranded DNA into a single strand, and then it's moving through the pore and is reading in real time this signal, right? And so that signal sometimes has noise and sometimes creates error rates. In the early days of nanopore sequencing, those error rates were around 25%. Now they're down around 15% on average. The great thing about it is it's small, right? So it has a high error rate, but here's a device. It fits in the palm of your hand. It can plug into a laptop. You can take it to the field. You can do real-time sequencing with it. The most notable case of real-time sequencing with the MinION device was tracking the Ebola outbreak in Africa just a few years ago, right? Where they were sequencing the virus in real time, whole genome sequencing, whole genome comparisons, and pinpointing some of the unique features of the genome that were contributing to the spread of the disease. When we look inside this device, what we see is the sequencing flow cell, which is made up of all of these different pores. There are 2,024 pores per flow cell, and they can move through the pore. A DNA molecule can move through the pore at about 250 bases per minute. So what we want to do is actually optimize all of these pores, right? And so on average, if we could occupy most of the pores most of the time, I meant to also mention, these are living pores, right? These are not some sort of static pore that can last forever. So one of the important approaches here is that your flow cell has to be fresh when you're ready to sequence. You don't want to buy your flow cell a month ahead of time and keep it in the refrigerator. There will be some degradation and you'll get a loss of efficiency. So we're interested in maximizing efficiency across all the pores on the flow cell. And we're interested in getting the best quality material into the flow cell as possible. And so our technical approach in this area was to use a pure culture, extract high quality DNA, and avoid shearing as much as possible. And so in my lab, one of the ways we avoid shearing is to actually pour instead of pipette. So we decant liquids instead of trying to pipette liquids, or if we have to use a pipette, we actually cut the tip off so that the orifice that the material is going through is larger. We sequenced on the nanopore 1D genomic DNA by ligation, flow cell 9.4. And then we assembled and polished, and then we evaluated our assembly quality using Busco. And I'm now going to go into more details about that. So I mentioned you want to occupy as much of your flow cell as you can. For those who haven't run one of these, when you do, you get an output. This is actually the summary output. You get an output in real time that uses the same color scheme. So here in light green are pores that are actively sequencing. Here in dark green is a pore that is getting occupied. And in blue is a pore that has sequenced and is recovering. In light blue is inactive. And what you really want is as little light blue as possible. On the x-axis is the time scale, and on the y is the state time equivalent percent. That's really, you can think of that as the percent occupancy of the pore. And so you see, first of all, it declines over time. This is out to 2 days. And so we can get our maximum amount of sequencing done over 2 days, but you can actually do a reasonable amount of sequencing within 2 to 3 hours, right? Here's the 1 hour, 30-minute mark. If all you were looking for were amplicons, right? Particular sections of sequence, that would be sufficient to get you plenty of data for detecting those sorts of things. So we ran it out for 2 hours. The expected yield on that data is about 4 to 5 gigabases per flow cell. Instead, what we got was 2 times that amount of coverage. So we got 10 gigs where we expected 5. No doubt due to having a really fresh flow cell, really high quality DNA, worked out really well for us in this case, right? So when we look at the data, we were also around 90% accuracy. So our error rate was around 10%. So we had lots of data, lots of pretty accurate data. The only disappointment was the mean read length, which was around 7 KB. I'd like to have had that higher, but what you can see from these dots is we had some reads that were up around 100 KB, right? And these reads are the ones that are really important, these 60 to 100 KB, where we can have repetitive regions that span 10, 12 KB. These reads can actually bridge those gaps for us, right? And we can assemble those complicated regions as a result. So schematically, what does it actually look like, and what did we do? So we generate these long reads, and some programs such as Canoe will try to error correct these long reads. And error correction for really, really noisy reads like this takes a long time. When I assembled this genome using Canoe, it took about 32 days just to get my error correction on the reads. Instead, Minimap is a program written by Hang Lee. And what Minimap does is actually just map these reads to each other, right, to find overlaps. And that gives us these sorts of overlaps that are, what I call uncorrected pre-assembled reads, and they're put together. So now we've gone from 7 kb and sometimes longer into these overlaps where the average was closer to 30 kb. Well, that makes a big difference now in assembling to a contig. Minia SM is another program written by Hang Lee, where now we can take those uncorrected pre-assembled reads and start stitching them together into a contig. And this is done largely through overlap, but the way Hang Lee has written the program, it uses overlaps plus a graph assembly. So not only are we able to utilize the power of overlaps, we're able to utilize the algorithm behind graphs to find where there are complicated regions, we can start to resolve them. And what we end up with is a contig that has the exact same error rate as our raw reads, right? We have not corrected for error at all. But now we can take a combination of short and long reads and go back and polish this contig to correct all those errors, right? We can count on the efficiency, the precision, 99.999% correctness of short reads to correct all of these sequencing errors in the contig itself. So we went through this whole process. This actually, in contrast to taking 30 days to get our error read, our corrected reads to then begin to assemble, we were able to do this in right around 72 hours of compute time. So a significant difference there. When we look at what we ended up with, we find, so here's the reference assembly again, 56 megs. When we just assembled the nanopore contigs as raw sequences, what we find is 94.3 megabase pairs, around 603 contigs. The 50% of our genome was contained in just 152 of those contigs, and the average length for 50% was 194 kb. So this was great, but it caused me sorry, let me go through the scaffolds first. So the nanopore scaffolds, scaffolding is taking those contigs and then trying to stitch them together into even longer reads by going back to the raw reads and seeing if we can find reads that bridge gaps in the contig assembly. And so here, it improved the numbers slightly for total length, fairly good for number of contigs, and fairly substantially for the amount of genome. 50% of our genome was in 95 contigs. So that means that we really bumped up the average length of sequence, right? And here we're at 313 kb, and you contrast that with 34, right? So we're close to 10 times that length. So I was excited to get these results, but I was also really scared because I didn't know why it was so much bigger, and I thought, oh, I'm extending all these haplotypes. The genome is not going to be of really good quality. And so I rushed up to John McDowell's office and I said, I'm getting a genome that's almost twice as big, and this is kind of scaring me. And John just laughed at me and said, oh, everybody knows there are more repeats in there than we can acknowledge. And so as a test, I went to the gold standard for Genome Assist, which is confirming the genome size with flow cytometry. And so here we developed a protocol to sort of more quickly be able to isolate nuclei, stain them, and check the size. So here's our Capcc sample. Here's Synengia speciosa, which is another genome that we recently assembled and done a lot of work with. So it was a good internal standard. This one's around 392 megabases. And this estimate for Capcc from flow cytometry was around 110 megabases. So I felt more confident that what we were seeing was real and that this assembly was good. And in fact, I was actually happier to have the estimated genome size a little larger than our assembly size, because I know from the data that we're still going to miss repeats. We're still going to collapse some things that we shouldn't. And so that fact gave me real confidence. And another way to assess our confidence in this genome is to look at something we call a BUSCO score. BUSCO is a acronym. The basic idea behind it is that we are looking at conserved orthologs across a wide array of organisms. So these are genes that we have really high confidence in their sequence, in their position, in the level within the organism. So we can compare that across multiple assemblies and get some idea of how good the assembly is. And so here's what we were talking about earlier. So this is the reference assembly. 91% BUSCO score is a pretty good BUSCO score, right? The Arabidopsis thaliana, the model plant, is around 94%. Tomato, another good reference genome, is around 92%. So, you know, when we said this was a good assembly, it is a good assembly. It's 0% duplication. Here, of course, duplicates were held out, right? The repeats were held out from this assembly, and that's by necessity. So if we just look at the raw long read assembly, what we see is that it's really bad, right? I mean, strikingly bad. I was expecting 70%. I wasn't expecting 48.7% complete BUSCOs and 50% missing, right? So that tells you what, even at 10%, what the error rate does to the quality of our assembly. But we can take our long reads and short reads and polish it. And I guess one of the reasons even though we have noisy data in the long reads, one of the reasons we really want to be sure that we use long reads and polishing is that the short reads still can't bridge the repeat gaps. The long reads can, and we have 100x coverage of the long reads. So we have really high confidence that we can correct. And when we do that, we see a significant improvement, but still not great, right? So we move up to around 62% total. And now we're starting to get better single copy orthologs, some indication of duplication, but we're still missing a lot, right? So the bulk of the genome is still uncorrected. However, when we just use 2 rounds of short read polishing, we're now in a high quality zone, right? Now we have 97.49% complete BUSCOs. So we're really sure that these gene calls are accurate. And we're really sure that this duplication rate is fairly close to accurate. And now we're down to just 2.2% missing. So we have high confidence that this genome is assembled well, the gene calls are fairly accurate, but it's important to remember at this point that any assembly really is just a hypothesis, right? So I think John made a great point earlier when he said, you know, the genome was the best it could be, and still there's room for improvement. So this genome, this one that we've done, is great, but there still may be room for improvement, and likely is. Now, genome assembly, just for genome assembly's sake, is pretty boring, at least to me. That's not, you know, I want to make genomes to do something with them. And it just so happened we embarked on another collaboration with the McDowell lab to actually use this genome to try to identify genes that are differentially expressed when Phytophthora capsicii is exposed to iron versus a depletion in iron. And so this experiment just had capsicii on plates that John Herlihy grew, replete and deplete in iron. They used single-end RNA-seq to generate the sequencing data. And then my lab took it and mapped it to the annotated draft genome and then conducted a DE-seq analysis. And you know, we have high confidence that our gene calls are correct, and therefore we have high confidence that what we see in this pattern is true. And of course, you know, this is a bioinformatician's dream, these data, right? Such clear separation between treatments. I don't know, it's like candy. So we're really excited, and we're going to follow this through and hopefully be done with this analysis soon. But it just shows that this genome is not only is it nice to update the genome for public use, but also for our own use. So in conclusion, we generated long reads from nanopore sequencing, and it really, really helped to resolve this highly duplicated genome. Aside from the cost of the nanopore itself, which, depending on what package you get, is somewhere between $3,000 and $5,000, we generated these long reads for just a $1,000 flow cell. And then we developed a pipeline, just putting together existing software that could actually resolve and assemble these noisy reads in about 1/6 of the time of other programs. And with that, we are happy to take any questions. This little show me the data. When I teach my genomics sequencing class, I used to show this slide all the time because there was insufficient data to really prove that nanopore was useful. And now we know for sure that it is quite useful.
Thank you. So I'll do the clapping. I'm sure there's some going on in the audience. That was really interesting, a lot of fun. I do not know much about genome sequencing, so that was a really interesting talk for me. So I see, if you have questions, please type them into the Q&A. And I see we have a lot showing up. And Lee Jun Ma, who is my co-host, and I will take turns reading the questions. Do you want to start, Lee Jun?
Absolutely.
Read off the first?
Absolutely. This is exciting. This is a question from Bangkok. And sorry, I probably doesn't pronounce your name correctly. Ira Pong. Anyway, the question is, hi, I'm from Bangkok. Is it possible to count the number of chromosomes, for example, by long reads?
Yeah, so not by long reads alone, but there are several sequencing strategies, I'll call them, to be able to get the genome separated by chromosomes and then sequence it and then reassemble it based on chromosomes. One of those methods is called Hi-C. Some of you may have heard of Hi-C or BioNano. So there are technical ways to do that. Yep.
Just to follow up on that a little bit, do we know the number of chromosomes for this particular genome?
I think the number of major chromosomes are known. I don't have that number off the top of my head. I think the question really gets interesting when you start considering many chromosomes, supernumerary chromosomes, whatever. Li Jun, you above all others understand how interesting those are. They've not been explored at all in oomycetes.
Let's work together.
And that is one of the goals of these, which is to connect the community. Okay, so I'm going to read off our next question, which is by Liliana Cano, who thanks Dr. Haak and said, what strategies are you using to handle or improve the issue caused by heterozygosity in the P-capcc genome?
Yeah, so that's a fantastic question. We skirted that issue by using this particular strain or isolate. However, one of the ways that we can do that, particularly with long reads, and what we did in this assembly that I didn't talk about, it's sort of technical issue. I'm happy to talk about it at some point, but with long reads, we can actually phase and get haplotypes. So these long lines of haplotypes that can, we can then start to resolve that heterozygosity. So initially in the assembly, we were around 130 megabases, but that was because of unresolved haplotypes. Once we resolved those, we actually dropped back down to around 95 megabases. So fantastic question.
All right, next question. So it's from David Livingstone Nisbah. Using nanopore technology, at what point does the DNA split into single strain? And just a follow up that question is, why not use a whole DNA double strained?
Great question. So the split happens right at the pore itself. The enzyme is actually bound to the outside edge of the pore, and it's split as it goes in. The reason we want to use a haplotypic strand is just from the last question, right? Heterozygosity. So we want to have a single side and sequence that and assemble that so that we're not trying to resolve differences between the strands in our assembly. That it makes the assembly much harder.
Okay, so the next one is from Roger Innes, who asks, for short read polishing, are you using Illumina to generate the short reads?
Okay, hey Roger, hope you're doing well. We did use Illumina to generate the short reads. However, the short reads were already generated. So one of the things I neglected to mention is we were trying to find a system to sequence. We were trying to see what bump we could do. And so in this particular, for capsaicin, but also for the particular isolate LT263, some data already existed, right? And so there was plenty of short read data that we could use to polish these long reads. And so we didn't have to generate that de novo.
Mm-hmm. So next question from Sirapong Krajanhi, probably didn't pronounce your name correct, I'm sorry. And this question actually from Bangkok. And the question is, what was the Buscore core gene set used to evaluate the assembly quality?
It was in the Strumenta files. I'll have to go back and look specifically. I don't remember. We tested between two different existing databases, but both of them were relevant.
Okay, our next question is from Sean Deller. Why did you choose this strain of P. capsaicin? And they're writing from Syngenta in the UK.
Great, yeah. So as I mentioned, we in going and looking for an organism, I wanted something that was, of course, relevant, but also where a bit of work had been done. And when I approached John about it, he was like, "Have I got the thing for you?" So, it was well studied. There were existing genomic resources that we could try to leverage to sort of thinking about the typical lab, right? A typical lab has been working with a particular strain for some time. They may have a bit of MySeq data or other Illumina data laying around. And if they could just add some long read sequencing to that, they could gain more information. And that really was our goal here.
I'll add to that a little bit. The reason that we use this particular strain is that it's highly virulent on Arabidopsis. Not all Phytophthora capsaicin strains have that attribute, but thanks to Francine Gover's work, we knew that LT263 grew very happily on Arabidopsis. So that's our lab reference strain. And as David mentioned, we had already committed to doing a lot of RNA-seq with this particular strain. So we knew that resource would be available to apply to the new version of the genome for more accurate annotation.
Great. So next question from Michael Said. And hey, thanks for the clear talk. I have a question on the Buscore duplicates. Assume that you succeeded in an haploid genome assembly. Do you think 20% is a real duplication? Maybe indicative for aneuploidy? Or do you think that some haplotypes are not flattened and represented in your assembly? Thanks, Michael.
I think yes to all of the above. So when we, in the paper, we dive into a little bit of evolutionary analysis. So when we look across the clade, what we find, especially from some of the long read assemblies coming out of Brett Tyler's lab, that we find that is relatively consistent, right? 15 to 20% duplication is not abnormal for the clade in which this particular Phytophthora sits. So I believe that we're right in somewhere north of 15%. Is it 20%? Are we collapsing some? I'm sure that we're collapsing some haplotypes, but no, we just need more data.
Yeah, I think I follow up because this is intriguing. A couple of questions connected with that. I thought one simple technology maybe you can use is looking for the sequence coverage across the whole genome. If Michael's hypothesis is true, you should see split of bimodal distribution of your coverage. If it's not, then you should more uniform distribution.
That's right.
Of course, for repetitive genome, you're going to see a bump somewhere else. But if you have two major haplotypes split, that could be indicated. I've just thought about that.
That's right. And so what we, when we assembled the genome, we actually went through and looked up at coverage across all of them, and then basically did a coverage correction for haplotypes where we thought we were actually collapsing.
And this next question is really, people are very interested in this. So the next one is by Andrew. I'm not sure if it's Gitto or Gitto. Andrew asks, can heterozygous alleles be kept in the assembly? So a lot of interest there.
Yeah, yes, as long as you have enough Illumina data. All right, so as long as you can be sure that that call is in fact correct, you can.
So follow up that question. I think it's a very interesting question. So we also worked a little bit on OMIC, and we are starting work on the RNA-seq data, John. So the haplotype really play a role there because we noticed that effector genes capture higher diversity, and that expression of the two haplotypes does not express equally. So get understand of two different alleles may be important. I don't know, we just started that. So that's why I guess caught a lot of fascination here. But okay, I read for the next question. It's from Valerie Williamson. So what did you learn about repeat sequence and distribution from the new assembly?
Yes, so the in terms of delving into the repeats to see what's there, that's currently being done as a project in my lab. Just sort of from a overall perspective, looking at KS plots, trying to estimate something about repeats and repeat evolution within this group. What we found actually is there was already circulating evidence to support a duplication leading to this lineage, and we found further evidence supporting that notion that was originally again put forth. So the repeats in some cases are very old, and in other cases, we're actively investigating to see what sort of effectors are in some of these newly resolved repeats.
Interesting. So Yufeng Fang writes, hi, to follow up, what was the coverage of the nanopore reads? I don't know if you want to answer that quickly, and then I'll read his other half question.
Yeah, so our coverage was about 100x.
Okay, and have you checked chromosomal elements, including telomeres and centromeres?
Nope.
I happen to know Yufeng's an expert in this area, so go for it, buddy.
Yeah, that sounds great.
Can I follow on that? Because when you talk about your algorithms through your introduction, and the Minimap too developed, it's very intriguing. I think it's a very clever process. But I wondered, did you test it with a minimal coverage requirement? 100x, it's a lot for the new sequencing. And I guess you said you're very lucky you have more than you anticipated to get into it. I just wonder in order to make that algorithm effective, it's a minimal coverage requirement.
Is there a minimal coverage requirement? Yes. Dr. Lee suggests that with nanopore, you have no less than 30x.
I see. Great, thank you.
Yeah.
Okay, so here's the next question from Ashwabi, anyway, the question is, what about the reliability of using nanopore sequence in sequencing the genome of a tetraploid, which exactly has 4 sets of chromosomes?
Yeah, I'll let you know in a couple of months. We're working on a frog right now. It's a hexaploid. The short answer is coverage, right? The more coverage you can get, the better you can start to resolve these things. But I think it will also entail some other techniques such as bio-nano, where we can actually try to pull down either telomeres or centromeres and try to really pinpoint chromosomes.
So Liliana Cano has another question. Thanks, Dr. Haak. Would you say nanopore sequencing could also be used to study sequence a collection of PCAP-CC isolates as well? Maybe in, maybe in that way, nanopore data can improve traditional DNA-seq Illumina data or make it cheaper.
Yeah, so I would actually suggest the opposite. Right? I would suggest that you use nanopore to generate your reference. If you're looking, for how things compare to a particular reference, use nanopore to generate the draft sequence that you want to use for that reference. And then you use Illumina to get all your population data where you can be sure that all the allele calls are in fact correct. And incidentally, still, that would be cheaper, right? Illumina is still per base is much cheaper than nanopore.
So next question from Juan Carlos Diaz. How many times one can use a nanopore matrix?
So that depends on how much sequencing you do. So if you do like what we did, which is use it for 2 days, 48 hours, it's done. If you use it for 2 hours, you can wash it and there will still be some viable pores on it. So the number of pores it uses is a function of time. And so that's the rule there. And in our experience, that was very true.
So it's just to follow up on that. Is a washing technique matters? You do 2 hours, and then you're washing, and then the second round. I was just trying to see from economic perspective. Do you suggest to say, yeah, do 2 hours, wash it, use it a couple of times, or no, do the 2 days and get the maximum you can?
I think it depends on your goal. Mm-hmm. So I was helping another lab that was trying to see if they could wash off a leaf surface and sequence that, and just like microbiome sequencing, just capture a particular gene and count to see what the abundance was there. Doing that, washing after 2 hours, that was actually pretty effective. They got 4 runs out of a single flow cell.
Makes sense.
Interesting. So our next one is, and we don't have a name up there, but somebody from India, who, and I just want to point out, we have had people from all over the world. Just to do a side trip into the chat, we've had Thailand, India, Bangladesh, and I know I'm missing some of the other ones. But yeah, oh, Nigeria. So people from all over, it was terrific. So this person asks, what strategy can be followed to sequence tetraploid plant species using nanopore sequencing technology?
Yeah, so we have a collaborative project where we're sequencing tetraploid plant that's got a 10 gig genome. And for that, we elected to use PacBio rather than nanopore, so a combination of PacBio and Illumina. And the reason we picked PacBio is because nanopore is somewhat temperamental, right? Especially with large plant genomes. So I say we got lucky and got twice the coverage we expected from nanopore, and that's really it. So, Lee John already alluded to the fact that with nanopore, you really want to have the best hands in your lab preparing the DNA, loading the flow cell, taking care of the flow cell. All those things add up to affect your coverage. And one day you can get twice as much as you expected. The next day you might get half as much as you expected. We tried to sequence a complicated parasitic plant genome on the nanopore, and the DNA quality numbers were just a little bit off, and it resulted in really low quality data. So it's really important for nanopore to have all those steps in a row. Now, do I think that you can do it? Yeah, I think people with molecular biology experience can go in and set up their flow cell and run things without problem. And if you do, if you do run into a problem, you can always get another flow cell and get a bit more data. But for a large sequencing project where you know that you're already going to be spending $30,000, $40,000 on long reads, PacBio still gets you more bang for your buck in terms of the error rate.
Right. So next question from a friend, and I think maybe this is better answered by Jean yourself. And the person is really interested to get free materials to understand sequencing molecular plant pathology.
That's a terrific question. And I can only give a little bit of the answer, but I'm hoping other people will jump in. There's a lot of information available on the internet, which is a great place to start. Certainly, this entire seminar series is free, so I encourage you to go back and listen to some of the ones that you might have missed and to tune in for the future ones, which are all going to be, I can't promise they'll all be on pathology, some might be on symbiosis, but they will all be on molecular plant microbe interactions. We also have, remember, all of our technical advances are currently open access, and all of our resource announcements. And we are, as of January, the whole journal will be completely open access. You'll be able to access anything going back and also going forward. And that's a great place. John, do you have anything to add? Any suggestions?
I was going to point out that a lot of the legacy review articles in MPMI, as well as upcoming review articles, will be either are or will be open access. And collectively, there's some great resources therein.
And as a new editor for the journal, I will also tell the audience that if your particular subject you wanted to learn, which you don't find available yet, you can write suggestion to the journal to see whether we can help you to find additional, at least we can point you to the right direction.
Very good suggestion. And that actually reminds me that 1 month from today, we're going to have, or whatever, not our next seminar, but our seminar after that. So 4 weeks from now, we're going to be having Brandon Reagan from Tessa Birch-Smith's lab is going to be talking about a really nice review that they wrote on plasmodesmata and plant virus interactions that came out in our January focus issue on the cell biology of plant virus and virus vector interactions. And that's going to be, we're opening that up completely open access as well. So that could be a great way to get some good background about the cell biology of those interactions. Okay, let's move on to the next one. We have Shruti Barathan, who is asking, what are the shortcomings of using nanopore sequencing?
Yeah, so I highlighted some of them, right? That the input material is really critical. The quality scores, many of you who are used to sending things off for sequencing, if your quality score is a 1.7, it doesn't really matter for Illumina. For the 280, 260. If for nanopore, it really matters. The closer to 2 you can get them, the better off you're going to be. The second big drawback is just the amount of material, right? We're talking about needing micrograms of DNA starting material as opposed to nanograms. So the starting material is another big one. And then again, care and handling of your flow cells is critical. When they come in, they've got to go right into the refrigerator. They can't sit on the bench for a while. So, all things that we're used to with enzymes and other things, but still, it's sequencing. So sometimes that's a barrier to get over.
So the next question from Ninofa Mirza from India. How to go about genomes where we have no knowledge about genome size or the number of chromosomes?
Yeah, so the number of chromosomes it can be important, but the genome size is critical, right? In estimating what coverage you need and all of that. And I really feel like the best way to go about that is using flow cytometry to estimate what your genome size is. You need a protocol for doing it. My lab is working on a protocol for oomycetes in general, and I'm happy to share that with anybody who wants it. But the key is having a protocol to be able to isolate nuclei and stain them, and some sort of control sample, and knowing something about your organism from the literature about its general size. If it's going to be 100 megabases, then you need something that's larger than that, right? So you always want to go, your internal standard should always be larger than what you expect your sample to be.
Interesting. So our next question is by Liliana Cano. Liliana, I'm going to hold off on your question for right now. You've had a couple of really good questions. Let's see. I want to get to some people who have not asked the question yet. So our next one is, do you want to read the next one, Li Jun?
Sure. This one probably, I think we talked, but let's touch on that. This one from Zhao Hui Liu. And the question is, can you please talk about the pros and the cons for nanopore and the PacBio? Thanks.
Yeah, so pros and cons, we can start with cost. Like the initial cost for PacBio is higher because on the sequel system, you can't just buy one small unit, right? You have to buy either a full smart cell or a cluster of smart cells. So right there, your job has to be big enough to justify $5,000 to $8,000.
Mm-hmm.
Whereas with the nanopore, you can buy an individual flow cell, you can buy 2 flow cells, you can have the same unit set up for doing long read RNA-seq data or genomic data, right? So it's really flexible in that capacity. Cons of nanopore versus PacBio is the error rate, right? The error rate for PacBio is somewhere around 3% now, so it's much better still than what nanopore is. The cost per base is lower with PacBio, but after you get over that initial investment. So both of them can give you long reads. The longest reads right now are coming from nanopore, right, where you can do all the customization. So recently someone did a full chromosome arm of yeast. So
very interesting. So our next one is by Gautam Shirsekar. Have you annotated this genome? And if so, could you briefly explain the strategy? That might be a whole nother talk, David. I don't know.
Yeah, I can briefly do it. We did annotate it. The annotation is available with the genome. You just follow the link from the paper. The strategy was, we used Maker to do so. The strategy was to first do machine learning based de novo identification of gene regions, and then go back with the Lamore lab did a beautiful job over the years of annotating and updating the annotation for the genome they published. So we used their data heavily to identify the true genes from the predicted genes in this genome. And then, as John mentioned, we also had in-house some RNA-seq data from this exact strain that we were able to also use to verify the predicted genes.
Just to follow up on that question, how many total number of genes is the genome have you annotated?
I don't remember. I'm sorry.
Is it complete?
I should look it up.
Oh, that's okay. So I can read the next question.
It was almost identical to what the Lemoore lab had identified. Maybe just a handful more.
So next question from Junia Yin. What strategy is used to arrange nanopore contact to scaffold? So basically the scaffold strategy.
Yeah, so it's the program that we used is called S-Space long reads. And essentially what it does is take any given information, any information you can give it via the raw reads or other Illumina data, and between when it's someone already alluded to in their questions, when we have contigs that get stuck together, what happens is all the gap space gets filled with ends. And what it does is goes and tries to overlap reads across those ends and fill in that information. And of course, you set the threshold of how many reads it's going to take and how big a gap it will fill to try to stitch those together into scaffolds.
So our next one is Nilofar Mirza. I'm talking about fungal genome. Want to ask about heterogeneous genomes more, if you can elaborate a little.
So the real problem with really heterogeneous genomes. So as Lee Jun mentioned earlier, that we had a project where we were sequencing downy mildew, right? And when we align and go and count our contigs and look for overlaps, we see two very distinct, very large bimodal peaks, which indicates a high degree of heterozygosity, more heterozygosity than humans, right? And so that's coming from other bits, right? There's more than just the nuclear genome there when we see that much differentiation. There's no great resolution for that, except for getting a lot of Illumina data and more high quality long read data.
Next question from Francine Gavers, and this is related with your RNA-seq experiments. Thanks for the nice talk. What is the concentration of iron you used? Do you see any phenotype upon treatment? And what is your thought about the biological relevance of iron in the interaction? You're muted, John.
The data that David showed was from comparison of P. capsese grown on iron replete versus iron depleted synthetic medium. And I don't recall the concentration of iron in the iron depleted medium, but it was quite low. I think on the order of 10 micromolar or so. Now, in terms of effects, when we grow P. capsese either on iron depleted synthetic medium or iron depleted Arabidopsis plants, growth is reduced, but not as much as you might expect, on the order of, let's say, 25 or 30%. So I take that as evidence that Phytophthora capsese is very good at scavenging iron from its environment. And one of the things that we hope to learn from the comparison that David was showing in the slide are the mechanisms through which it does that. And one of the nice things that David's analysis has uncovered is a putative iron scavenging regulon, which indicates that P. capsese has a mechanism for active transport of reduced iron from the extracellular environment. That may not mean much to you if you're not an iron nerd, but it possibly contrasts with other plant-associated microbes who rely predominantly on secretion and then uptake of siderophores as their mechanism for collecting iron. So there's a lot more on that to come. In terms of its overall relevance, obviously Phytophthora, like every other organism, needs iron to survive. And so, as I said, we're interested in the dynamic between the plant and the pathogen in terms of fighting for this iron. And I think the question's particularly interesting on the plant side because, as you know, plants require iron as a critically important factor in the immune response for generation of an oxidative burst. So is it indeed actively trying to withhold iron at the infection site? And if so, is there a trade-off for immune efficiency? And what are the underlying mechanisms through which all of this occurs?
Interesting. So that was interesting to get into that bit of iron biology. And just before I read the next question, I want to point out that we have posted in the chat a link to the actual paper. So it doesn't have the iron part in it, I think, but that's new stuff that you got added just because you came to the seminar. But you can follow that link to get lots of the details from the methods, etc. And if you return to the site where you registered for this talk, you'll be able to listen to the recording once we post it, which will hopefully be in the next day or two. Okay, Howlin Yu says, thank you for the very informative talk. Comparing with the non-repetitive genome part, what other distinct genomic features besides repetitive are for those repetitive sequences that are assembly?
Yeah, it's a great question. Again, I have a collaborator whose expertise is in repetitive gene family evolution, and hopefully he's working on that analysis as we speak. So we don't know yet is the honest answer. I suspect that we're going to find that there are some repetitive housekeeping type genes in there, and certainly some effectors are going to be found in this enriched repeat region.
Yeah, for those of you who aren't familiar with omycete genome organization, effector genes tend to congregate in regions of the genome that are repeat rich, and that mode of organization is thought to facilitate rapid evolution with the host. So what I would expect are that there are quite a few interesting and heretofore unidentified RXLR effectors and other types of effectors within this new fraction of the genome.
Right. So next question from Julian Ying. Does a polishing process decrease SNPs?
It's a great question because it can. And so you have to be really careful how you go about polishing. So we were fortunate that the reads that were available, the short reads that were available, were from this isolate, and in large part represented this isolate very well. If you take your reference assembly that you're making and you use reads from something else to polish it, yes, you will polish away polymorphisms that might be important if you're looking to do population genomics. So I would advise against doing that. In our case, the reads that were available, of course, because we chose this organism in this particular strain, were from the same organism. So if you, I saw that there was another question that I could go ahead and answer, so could you use this approach for a new organism? And I wouldn't, right? For a new organism, I would try to figure out how big the genome size is, and then I would use a combination of long reads and short reads to try to build my own reference for that organism.
Okay, I'm going to jump down to the bottom because that's been put out of order because of time zone. Alfredo Reyes Tena says, from Mexico, how much does the MinION cost?
Yeah, so the device itself, I think if you pay $3,000, you get the device and a single flow cell If you buy the starter kit, I think it's like $5,000, and you get the device, you get reagents, you get 2 flow cells of your choice. Once you've paid for that, then you just buy the flow cells afterward. And the flow cells are regenerated by Oxford Nanopore. So you get the flow cell, you use it, you wash it, and you ship it back, but then you buy another one.
So next question is on BR3. I think again, it's a strategy. Maybe you already touched, but I'll read just anyway. Thanks, John and David. So you rely heavily on the short reads database in polishing. Does that mean this method is not suitable for assembling a new genome? So I think you touched. If you want to skip, that's okay.
Yeah, I think that that was covered in the last question. Karina Atristan, when you map the RNA-seq reads to the new genome, are you improving the percentage of mapped reads with respect to the reference genome? And at genome prediction level, did you improve this parameter?
Yes, to both questions. So we saw a dramatic increase with the proportion of RNA-seq reads that mapped. We went from around 80% to 94% reads mapping, which is a nice big change.
So next question from Juanita Gill. What was the coverage of the Illumina reads used for polishing? Where you recommended a minimal coverage to polish or draft the genome from nanopore reads?
Yeah, so the minimum recommended coverage is somewhere around 60x. We had available to us somewhere around 200x.
And, oops, sorry, it moved. Someone, we don't have a name for this one. How efficient will nanopore sequencing be in the case of metagenome study, especially rhizosphere microbiome? And I know you're trying this with microbiome stuff as well.
Yeah, that's right. So in our hands for metagenomes, it is fantastic because we can multiplex up to 12 samples. We can get really good coverage and long reads. 7 kb for a metagenome read is great, right? That means we've captured 90% of the possible full-length genes in the sample. And so I think it's going to be good. Soon I'll have some data to compare PacBio and Nanopore.
Interesting.
Next question from Marcelo. And actually the question basically is, can you use the same strategy to P infestans? Have you thought about it?
Yes, it could be done, and to my knowledge, it is being done. So, but not by me.
And Liliana Cano has another question. Would you say that the DNA input to be used for nanopore sequencing should have high molecular weight, same as required for PacBio sequencing? And would you recommend greater than 20 kb? So these are some specific questions. And also DNA extraction kit of preference. Sounds like people are out there pretty interested in trying your approach, John and David.
Yeah. And I just to tack on to something you said earlier, Jean, the fantastic thing about having the article open source is the link to some of these resources for some of these questions is available within the article, right? So we've posted our protocol, we've posted all the ancillary data there. And so in this open source world, I hope that people can use those resources. I would say that the amount required is more than for PacBio, and the higher molecular weight you can get, the better, right? And greater than 20 kb. So we ran a gel, we picked only the stuff that came at the top of the gel, which was closer to 40, 50 kb, right? So there's not a ladder. You just gotta go, you get your 20 kb ladder and you run everything out. Everything that's above that ladder, you take.
So I want to just jump in here and say that one reason that MPMI has made all the technical advances open access is because we really want you guys to try this. So we would like you to take advantage of this knowledge and see what you can do with your own projects. Okay, next question. Juan Carlos Diaz-Ricci says, wonderful news and many thanks. You want to go to the next one, Li Jun?
Oh, sure. So next, friends, which didn't put the name, but very interesting, bring back to the biology. We did the genome sequence. What are the next steps that would help us to elucidate why this pathogen species have such a broad host range? Could this be related to the duplication of the genome?
So I can speak to that one for a minute. I think it's important to make the distinction that all of the strains within the species collectively have a broad host range, but individual strains of P. capsicum likely are more narrow. Now, with that said, these strains are in many cases likely to colonize multiple hosts. So for example, the strain that we use for Arabidopsis, I think was obtained from a cantaloupe in East Tennessee or a watermelon or something like that. So to speak to your question, that's one of the big questions in the MPMI field. Why is it that some pathogens have a broad host range and others are restricted? I would, as an effector biologist, my natural tendency is to explain everything in terms of effectors, right? As either promoting or restricting colonization on a particular host, but that's undoubtedly a simplistic answer. And I think probably the answer depends on the nature of the pathogen in question and the host that it either inhabits or doesn't. Sorry for that non-answer, but ask it again in 10 or 20 years.
So our next question is from Sora S. Can we conclude that for large-sized genomes, we need to consider multiple platforms to get a complete sequence?
Yes.
The next question from Nilofar Miraza. How much processing capacity was required for this assembly?
Oh, that's a fantastic question. What I assembled this, or Chen Ming assembled this on our small cluster, which is just 96. So it's 48 processors, dual core, so 96 cores. We had available 1 terabyte of RAM. It only used about 150 gigs of RAM. So it's not, many people have gaming computers that are more resource intensive than what we used.
So the next one is also, we have no name. It says, thanks for a nice talk. And I think this is directed more towards John. Does the pathogen produce biofilm? In some bacterial plant pathogens, some biofilm-related regulators seem to also influence iron scavenging. Could that be the case in P. capsicum?
That's a really thought-provoking series of questions. What we know about P. capsicum, and I think this is for Phytophthora root pathogens in general, is that they tend to prefer penetration in the zone of elongation. Okay, so they congregate in that region of the root rather than being distributed across the entire root length. We see this really clearly with our pathosystem because we grow it in a hydroponic system, so it's easy to see where the preferential sites of attachment are. Now, what's really cool about your question is that it caused me to recall a talk that I saw at an oomycete meeting last summer where there was evidence presented that this congregation, if you will, has biofilm-like qualities. So I'm going to think about that some more. And for sure, there have been hints in the oomycete area of evidence for quorum sensing in the past, but no really coherent picture has emerged yet. And I think these are really cool things to think about. Thanks for bringing that up.
I'm going to jump in because that's interesting to me, because right here we have, we're thinking about Phytophthora is obviously a pathogen, but really this is an issue. This whole idea of trying to get iron is a key issue for plants, for microbes that are interacting with plants. And so recently in the Rhizobium legume literature, there's been a lot of excitement because people have identified 3 different iron transporters that appear to be used in the infected cells probably to deliver iron to the vesicles that contain the bacteria. So this whole, obtaining it from the environment, and when your environment is the plant, how do you obtain it? So it's a really interesting and extremely important question.
Yeah,
okay.
That's a big reason why I'm drawn to iron in particular,
Yeah.
is the implications for all of the other plant-associated microbes.
Yes, it's generally very relevant. Yeah. Okay, I'm going to read this second to last, and then I think we're down to the last question, Lee Jun. So Devduta Deb says, thank you, David and John, for the talk. How syntenic is the current P-capsicum assembly with other known Phytophthora species? And I think you have a very beautiful figure in your paper, actually.
Yeah. So there's strong synteny between all the members in this clade. There's obviously some repositioning, some rearrangement, but very little evidence that there are lots of gaps, lots of new genes within this clade.
All right. So we come to the last question. So it's technique. It's related with the sequence coverage. So I just read straight out from Ethan Fung. And a quick question, David. Generally, do you think long reads definitely produce better genome assembly? You said you got 100x coverage by just using one single whole cell. Do you think you get sufficient long reads? In other words, do you expect more long reads could result in lower sequence coverage?
Yeah, so it's an interesting question. What I first say is that the coverage has more to do with our error rate than the read length. I do wish that we had gotten an average read length closer to 20 kb. So in some pure PacBio stuff that I've done, I've gotten around 17 kb, and I had hoped for something like that with this nanopore data, because I think that would help resolve more repeats and get us a more accurate genome. But in terms of coverage, I think with nanopore you cannot sacrifice the coverage because of the high error rate.
Well, thanks everyone. I'm going to jump in here. We have answered, perfect final question. The final question from Nilo Farmirza. Thank you for this talk and for the lecture series. I would really like to follow the series. Where can I follow that? And Ashley, is it possible for you to paste in the link to our webpage? Hopefully we can get that and paste it into the chat. But you can always go to, and here she has done it. Look at the chat, and she has the link to our virtual seminar series. And if you go there, what you'll notice is that our next talk is going to be in 2 weeks. We're alternating time zones. If it's the wrong time for you, you can always come back to the same page and find the recordings. But I wanted to thank everybody for joining today. We had a really terrific audience. Thanks to David and John for a really interesting presentation and a lot of stamina, especially for David with the questions. I want you guys to notice that he answered questions for twice as long as he actually gave his seminar. So, so what that means is that we really got to interact and connect with a lot of you around the world. That was a lot of fun, a lot of good ideas. I can tell from the questions that many of you are thinking of trying this yourself. And I think that David and John would encourage you to do this, because this was, of course, why they wrote their technical advance. So please go take a look at the paper. It's open access, and it has all the details, the DNA kit, everything you want to know to repeat this. So I hope you'll share the news about this series, and that you'll join us in 2 weeks for the seminar by Hong Lu from Feng Cui's lab, talking about this tritrophic interaction virus, aphid plant, how a symbiotic virus helps out its aphid host and not the plant. Anyway, David and John, do you want to have any last words here before we close the session?
I'd just like to thank the audience for some terrific questions, and encourage you guys to reach out if questions occur to you after we sign off today. Just hit us up.
Yeah, Absolutely. I'll just echo that. I really enjoyed the questions. So that was fantastic.
Yeah, you guys did a terrific job. Okay, thanks everyone. Goodbye. And I hope we'll see you in 2 weeks.